Can you hear me in the back row? Great. So he's getting us going, and he's actually starting this back up again. This is just proof that science keeps surprising us. This was a headline in The Guardian today. They were looking at the complicated organic chemicals in the center of the galaxy, and how could I resist a headline like that? So if you ever wonder what's at the heart of the galaxy, raspberries and rum. So any questions from our chapter on RNA that you want to ask before we start thinking about protein synthesis? Any questions on protein synthesis? Just a reminder that today at 5, the review session will be following on after the class. If you'd like to work with Eli and AJ to review the material for the 4th exam, you're in the right place already. So we've talked about 2 of the 3 kinds of macromolecules that are kind of at the core of life, a lot bigger than ethylformate. We've talked about DNA replication and repair. We've talked about RNA synthesis and its processing. And we talked a little bit about how maybe RNA by itself can have some interesting catalytic properties. Is that still okay? I was echoing a little bit. Okay. But the majority of catalytic activity nowadays, of course, is through proteins. And so we need to figure out how to translate the linear information of RNA into the linear information of proteins if we want to actually get the maximum content out of DNA. And that's what we'll be exploring for the next couple of days, is protein synthesis and how that happens. Some of the key things to keep in mind when you're thinking about protein synthesis is that making a polypeptide, it's a polypeptide. There's going to be a cycle of repeating reactions to make each peptide bond. So for every peptide bond in the polypeptide, there's a little cycle of reactions, and then it moves on to the next one. Another aspect of protein synthesis is that RNA plays multiple different kinds of roles in making it happen. So there's a coding information or a translating role for the tRNA that can take nucleotide triplets and turn it into amino acids. There's a sequence role for the messenger RNA, lots of different messenger RNAs so that each cell can make 1,000 or several thousand proteins. And then the big, bulky ribosomal RNA that accounts for most of the RNA, providing the structural support for ribosomes, and it turns out, catalytic activity. Another intriguing thing about protein synthesis is that GTP plays a very big role in it. We've seen ATP do lots of different things along the way, but keep your eye out for all the different places where GTP comes in and is essential in it and helps it to go, in fact, in one direction. Protein synthesis is not the end of getting a protein to be useful. Simply synthesizing it would not be enough. You have to have it fold. And you remember from our earlier discussions in the semester that proteins in general have tertiary structures that are compact folding where everything's tucked away. Some proteins need processing of various kinds. We saw with chymotrypsin that it had to be proteolytically cleaved to be fully active. Some proteins even need to be exported outside the cell. And so the cell has machinery that can track that and process that and make sure that all the right changes are made and then it's shoved on out. The great majority of polypeptides are made by the processes we'll be talking about. There are a few that are made otherwise. This is a polypeptide antibiotic. But this polypeptide, although it is clearly a polypeptide, those are peptide bonds, nonetheless was not made by protein synthesis. It was made by an enzyme complex that had a separate enzyme for each addition in the structure and then one to close the ring at the end. So in general, the peptide antibiotics are the consequence of some pretty special machinery to make them. Now, the reason that change is needed, or a way of putting them together is needed, is that we're trying to match up 2 different kinds of things. We've got a linear array of nucleotides with 4 possible different bases, A, U, G, and C in the RNA. And then we've got 20 amino acids that we want to put together in a linear sequence. And in thinking about the fact that there are 20 amino acids and there are 4 bases, Francis Crick was able to figure out that there had to be a triplet code. And this is what he meant by that. if you think about a code that has 3 positions, where you're going to look at 3 nucleotides at a time, you could have any of the 4 in the first position, so that's 4 possibilities. In the second position, again, you could have any 4, so that's 4 times 4. You've got 16 now. And you could have any of the 4 in the third position. That'd be 64 possible combinations for a triplet code. So you have more than enough for 20 amino acids. If you were going to use 4 bases and only have 2 positions, you'd have 4 times 4. You'd have only 16. And 16 obviously can't code for 20 amino acids very precisely. It can only do it in kind of a sloppy way. There are some other aspects of the triplet code that were not immediately obvious, but were determined to be true by looking at the actual code as it works in cells. And we speak of the code as being sequential and non-overlapping. So by sequential, we mean you're going to read three triplets as one codon. You're going to move to the next base and read 3 nucleotides as the second codon, and you're going to move and so forth. So it's read in sequence, 3, and the next 3, and the next 3, and those 3 are not overlapping. So you read these 3 and then go to the next one. You don't use the last one as the beginning of the next one or something like that. Those were logically possible, but they were not practical, not terribly practical, and they turn out not to be the way it is. So those are both important properties of the genetic code as we use it. Another property is that it is unambiguous. If I tell you a 3-base codon, you know with absolute certainty what it codes for. It is utterly ambiguous. If I give you an RNA sequence, it is utterly ambiguous what protein is going to be coded for in that RNA. RNA to protein goes in one and only one mapping to the protein sequence. The code is, however, what's called degenerate. And this doesn't mean that the genetic code hangs out in the bars on Friday night. It means that if you look at a protein sequence, you don't know what the RNA sequence is. You can make guesses at it, but you don't know for sure. Because for this protein sequence, there are several different possible RNA sequences that would also give that specific protein sequence. So it's unambiguous going from RNA to DNA, but it's degenerate going back the other way because there are 64 codons and only 20 amino acids. They could have just ignored 44 of the codons, but that would have had problems of its own. Another feature of the code that was not initially completely appreciated, and the more we think about it, the more we appreciate it, is that it's a robust code. And by that we mean this. There are these 64 combinations. Some amino acids have more than one codon that codes for them. That's why it's a degenerate code. But they tend to cluster. The codons for any given amino acid are similar to each other so that if there is a mutation, there's a decent chance that it will not change the protein radically or perhaps at all. And we'll look at that in a little more detail on the next slide. Another aspect of the triplet code is that the frame matters. And what do I mean by that? Well, here we have a sequence of bases, same sequence on all 3 of them. In the top one, we're reading it starting at the A, 3 at a time, sequentially, non-overlapping. In the second one, we started at the C, 3 at a time, 3 at a time, 3 at a time. And the third one, we started at the second C, 3 at a time, 3 at a time, 3 at a time. And because it's unambiguous, we can tell what those different frames of reading would mean. In the first case, it would be threonine, isoleucine, serine, arginine. If we start at the C, it would be proline, serine, arginine, glutamate. And if we start at the second C, it would be histidine, leucine, glutamate, serine. So the sequence of protein will change depending on which frame you choose to read that messenger RNA in. We're going to see that it turns out to be an important part of protein synthesis to pick the right start site. Because when you pick the start site, you pick the frame, and that's going to determine how that sequence is read out. So there has to be some way to turn nucleotide sequence into amino acid sequence, and that's one of the challenges we'll be looking at. This is a little bit of a more detailed table showing you how robust the genetic code is. So take an example. Here's leucine. All 6 of these codons, all 6 of these triplets code for leucine. So if you know that the first 2 letters are CU, it doesn't matter what the third one is, it's going to be leucine. So you can have mutations in that third position, and it will not change the protein at all. Here are two more codons that code for leucine. If you mutate that A to a C or a U, you're going to get a phenylalanine. Well, that's not the same as leucine, but it's a lot more like leucine than, say, arginine is. So you have mutations that produce synonyms that are identical to the original codon, and you have mutations that can produce similar things, conservative changes. So these are all isoleucine in here, and you can mutate those into a valine fairly easily. And valine and leucine are often compatible within a protein. So this is what's meant by saying it's robust, that the organization of the codons mapping to the amino acids is one that many mutations are less damaging than they would be if the genetic code was completely randomly assigned. An important property. But we still have to find some way to turn sequence into amino acids. And that is the job of transfer RNA. Transfer RNA transfers the amino acid to the growing chain. But it's the adapter. It's the adapter that can match up codons in the messenger RNA and amino acids. And tRNAs always have a general kind of feature like this of having RNA structure with quite a bit of double-stranded areas, what are called hairpins, 4 major loops. This is a variable one in length. They have ones we're going to talk about in a second. But if the part that makes it match up to the RNA is down at the bottom in what's called the anticodon loop. So there's 3 bases at the bottom of the anticodon loop, and those 3 bases are going to be the ones that hybridize by Watson-Crick base pairing to the messenger RNA. They contain an anticodon, 3 bases that are complementary to, I should go like this, complementary to the codon. So they're going to pair up anti-parallel, just like double helices form, to match up. We say that a tRNA is an isoacceptor tRNA if it carries the same amino acid as something else. So for something like leucine that has 6 codons, the cell might have a number of different tRNAs. If they all code for leucine, they're considered isoaccepting. They all accept leucine. Now you noticed at the end there about how the terminal position could vary. Some tRNAs can actually hybridize to more than one codon. Obviously, they're only going to hybridize to codons that code for the same amino acid. But that, again, affects how many absolute tRNAs you need to have. Because of this ability, in bacteria, there may be only 30 or 40 transfer RNAs, even though there are 64 codons. And that's because some of those transfer RNAs are covering more than one codon. In eukaryotes like us, there can be 150. What exactly the purpose for the different tRNAs might be will vary with the cell and so forth. tRNAs are fairly small RNAs, 60 to 95 nucleotides about. A lot of modified bases in the tRNA. You may remember during the RNA we talked about RNA processing. A lot of secondary structure. So these are all domains of secondary structure where there's base pairing to create double helix, right? And these are the 5 major features. You have an anticodon loop. Opposite, at the other end from it, you have the acceptor stem, and the acceptor stem is going to accept the amino acid. You have a D loop, and you have a T pseudouridine C loop, and that's because of the composition and modifications in those loops. And then the 5th loop is variable and has different sizes and so forth. Those 5 major features pack up. This is the tertiary structure now of a transfer RNA. You notice that when it's all folded up, it's fairly compact. It's not sticking out all over. It's kind of slender side to side, so it's fairly long in this dimension. But if we were to look on it sideways, it's fairly narrow in thickness. And that allows more than one tRNA to fit side by side on the ribosome. Again, many of the bases have been modified. And in addition, there's a lot of non-standard base pairing. So there's some of it's Watson-Crick base pairing, but some of it's some of those non-standard base pairs we talked about in the RNA thing. But a lot of stacking. You can see it's highly stacked. Many of the bases are stacked. So most of the bases are, in fact, out of contact with water. They're in their base pairing, either standard or non-standard. It also means that the anticodon end can be a long way away from the acceptor end. It's almost 75 angstroms from the anticodon out to the amino acyl end where the amino acid's been added on. So it separates the coding aspect from the protein aspect just about as far as you could for that amount of mass. Now we have to get an amino acid onto the tRNA. That process is called tRNA amino acylation. So we're going to amino acylate it, add an amino acid group to it. And as you can guess, it wouldn't do that spontaneously. It takes energy. So here's one place where we do use ATP in the general domain of protein synthesis. Here's an amino acid and an ATP. And through the activity of enzymes, we make an amino acyl adenylate. So here's the amino acid part that's now has an AMP part here. So this is amino acyl adenylate, or amino acyl AMP. That, of course, looks like a high-energy bond. You can recognize it. So a tRNA can come in and replace the AMP, and now we have an amino acyl tRNA. And you can guess that that bond there has some energy kind of packed in there that we can use down the line. This is just showing you that detail in a little more. This part is the tRNA here, the adenine base. It's always on an A at the end of the tRNA. And here's the 3' hydroxyl is the place where the amino acyl group has been parked in time. So the enzyme that can accomplish this is called an amino acyl tRNA synthetase, or an ARS, amino acyl ribosomal synthetase, RNA synthetase. And its job is matching up correct amino acid to correct tRNA. It does cost us 2 high-energy bonds every time we charge a tRNA. And there is an amino acyl tRNA synthetase for each amino acid. So you might have an amino acid like leucine that has 6 codons. There is only one enzyme that charges all those tRNAs. So there's an amino acyl tRNA synthetase per amino acid, but it might handle 2, 4, 6 different tRNAs. It'll handle all of the iso-accepting tRNAs. And you think about how does it do that? And here's a picture that suggests how it does it. So here's your tRNA here, and here's your amino acyl tRNA synthetase. And it's just kind of hugging it all over. I mean, this is more contact between the enzyme and substrate than almost anything else I know of. And somehow or other, someone once described it as the synthetase sniffs the tRNA. But it can somehow or other identify all the tRNAs for a given amino acid and charge them with that amino acid. You can see it's a long enzyme. There's different parts of it that can detect the codon and can catalyze the addition of the amino acid at that CCA tail on the acceptor end. There are even some amino acyl tRNA synthetases that can proofread. So the isoleucine tRNA synthetase can filter out larger amino acids at the charging site where it's adding the amino acid to the end of the tRNA. And there's a second site that can detect whether it's as big as an isoleucine should be, and if it's not that big, it'll hydrolyze it because it's not big enough and start over again. It would rather sacrifice the 2 high-energy bonds than put in the wrong amino acid. So then we're going to use these tRNAs to read the genetic code. And this always gets a little bit hairy for people because nucleic acids and proteins are polar. They have a handedness to them, a directionality to it. So hang in here with us and we'll try to make sure that it's clear at the end of all this. 3 bases of the transfer RNA are going to hybridize to 3 bases of the messenger RNA. They always hybridize anti-parallel. That's how nucleic acids hybridize. But when we write nucleic acid sequences, we always write their sequence from 5' to 3'. So if you have a codon UUC in the messenger RNA, it's going to hybridize to an anticodon that would be an A opposite the U, an A opposite the next U, and a G opposite this. But the G here is at the 5' end of the anticodon, so we write the anticodon GAA. And I think this almost might be easy. This is one point. Sorry for the people watching the movie. This is one time when seeing it done dynamically may help. So I'm going to see if we can make this work. So if we have C, that's 5 prime to 3 prime. Then we want to go, it's going to be a G, A, but nucleic acids hybridize antiparallel like that. And so when we write this sequence, we write it 5 prime, G, A to 3 prime. Okay? I hope that's clear. You may want to do 2 or 3 just to fix it in your head. It's not terribly intuitive, but it will make sense if you do it a few times. In the codon, when we looked at the genetic code, we noticed that the third position of the codon could vary. I mean, that you could have any of several different ones mean leucine. There are some cases where one tRNA will read more than one codon. That would be the first position of the anticodon, because it's the first position of the anticodon that's opposite the third in the codon. And some of these tRNAs that can wobble, as we say, have an I, an inosine in that position, because that one can hybridize to more than one base. And so we can see tRNAs reading as many as 3 different codons, but they'll always be isoaccepting, coding for the same amino acid. And this is showing you this again showing you this in fair writing. UUC, C hybrid to G, U to A, U to A. This is written 5 prime to 3 prime, UCC. When we read the top one, it has to be read GAA. So, and this is several examples for you to work through for yourself. Well, that explains how we can match, or how we can map, maybe map would be a better word, particular codons in the RNA to particular amino acids. But we still need some machinery to accomplish that. The tRNA establishes, which codon is going to be which amino acid, but it doesn't do anything about getting them together. And to do that, we need the ribosome. So the ribosome is like a machine that can put together transfer RNAs, codons, and then sense that it's in the right place and then make a new peptide bond. That's the job of the ribosome, to match those things together and make bonds. When we look at a ribosome, this is still to me amazing that we know the structure of ribosomes, but there it is. Here's the 30S subunit of the ribosome. This is from a bacterium. And you can see, I think, in this that it's really quite an intermixture of both RNA and protein. The RNA in this case is gray and the protein is purple. There's lots of little ribosomal proteins sort of scattered all through the RNA. Both the protein and the RNA are important for the whole process. We're going to identify a bit along the way which ones might be more so at some than others. 80% of cellular RNA is committed to the job of making ribosomes. So it's a big commitment of cellular RNA. Eukaryotic cells have slightly larger ribosomes. They're 80S instead of 70S. S here is for Svedberg's. It's a measure for how fast things sink in a centrifuge. But just think of them as sort of like mass, if you like. They're bigger. Ribosomes, from wherever they come, have 2 subunits. They have a large and a small subunit. In the large subunit is either 50 or 60S, depending on prokaryotes versus eukaryotes. The small subunit is 30 or 40S, depending on prokaryotes versus eukaryotes. That is a little bit of a simplification. Red algae are one of the more primitive organisms. They have 75S ribosomes with 55S large units and 35S small subunits. And blue-green algae have 75S ribosomes and 55S large ones and 35S small ones. I'm not going to make a question on it, but I just think it's interesting that we see this point of molecular contact between the most complex of the prokaryotes and some of the most primitive eukaryotes. The large subunit needs at least 2, sometimes 3 RNAs to make it all work. The smaller subunit just has a single RNA in it. So the RNA is what provides the overall structure of the subunits. And then the proteins kind of pack into it. This is a kind of a cool diagram. Here you see the whole ribosome together with the RNA in the gray and the protein in the gold now. And what they've done in the diagram next to it is to show you just how extended and unfolded almost the little pieces of protein are. They're almost like the caulking on a window, the proteins are. So they coat aspects of the surface. Some of them kind of wiggle into the interior. The large subunit, the 50S subunit, is somewhat more solid, not as flexible, and it has a protein-free face where it interacts with a small subunit. Now, as you can imagine, the subunit interface, the place where the two subunits of the ribosome meet, is a very highly conserved aspect of the ribosome. To put together two things that big, you need to do it in a particular way. But it's also the place, that interface is also the place where the messenger RNA and the tRNA are held during translation. And when we look at the intact ribosome, we can identify, broadly speaking, 3 tRNA binding sites. And this is showing you a ribosome that's fully occupied by transfer RNAs. There's an A site for the aminoacyl tRNA. There's a P site for the peptidyl tRNA that has a peptide chain growing on it. And then there's an E site, an exit site, where the ribosome that's leaving stays for a brief time before it goes away. The 30S subunit, the small subunit, has the job of sort of checking or matching or providing a good environment for the codon and anticodon to match up. So it's exposing the RNA in a way that the tRNA's anticodon can get at it. And then remember how long a tRNA is. Up at the other end, the aminoacyl part is the large subunit has the job of making the bond to move that growing chain over. So there's a separation of functions here. The small subunit's kind of involved in the RNA side of it. The large subunit is involved in the synthetic side. We've talked before about how for any of these big processes that go on for a long time, you need to start it, you need to continue it, you need to end it. And so there's always a question of initiation, how you get started. And interestingly, translation always starts with methionine. All proteins, when they're born, have a methionine on their beginning position that may be processed off, but that's the first amino acid when the protein is synthesized. And so AUG, which is the codon that codes for methionine, is always the first position of the reading frame, the sequence in which you read out a messenger RNA into protein. Now, in prokaryotes, there is an area about 10 bases up called the Shine-Delgarno sequence. So you can see here that there's a short stretch of hybridization between the messenger RNA and the ribosomal RNA that allows for those to sort of be stabilized there long enough for this first AUG, this start codon, to be used as the start for the new protein. Both prokaryotes and eukaryotes have special initiator tRNAs. They code for methionine, but they're a special methionine thing that's an initiator tRNA for just that first amino acid that's the methionine. And a further modification is that in the prokaryotes, that initiator methionine is modified with a formyl group. So this is a formyl group that's been added on to the methionine to further distinguish that growing chain. So how do you put this whole thing together? All right. So you need a lot of parts. So here's a small subunit, a free 30S small subunit. And in this case, we're looking at prokaryotes because we're looking at a 30S subunit. We're looking at formal methionine. And to assemble all the pieces, you're going to need a ribosomal subunit. You're going to need messenger RNA, and you're going to need an initiator tRNA. And so the small subunit, the 30S subunit, provides a location on which the messenger RNA can bind, and this complex with the initiator methionine can sit down. But it doesn't do it on just sort of spontaneously. It has to be accomplished by the use of initiation factors. So IF here stands for initiation factors. These are for initiating protein synthesis. IF3 had provided us with that empty 30S ribosome. IF3 could split up ribosomes into their parts. IF1 is going to sit and block the amino acyl site so that it doesn't get occupied. And IF2, this particular initiation factor that has a GTP bound to it, can bind to the tRNA FMET, the methionine, formal methionine. So IF2, when it's occupied by GTP, can pick up a formal methionine tRNA. It's already been charged by the amino acyl RNA synthetase, and deliver it to the P site. So the A site is empty, and IF2 is delivering that initiator methionine to the AUG that we're going to start translation from. Now, this GTP molecule is going to hydrolyze, and when it hydrolyzes, IF2 comes off. And when it comes off, that frees up the possibility of the 50S subunit coming in. And so the consequence of the contribution from IF2 is we now have a fully assembled ribosome. It's got a molecule of RNA bound to it with the AUG site at the peptidyl site of the ribosome, and the A site is empty and waiting for something to happen. So now we have a charged ribosome ready to go. In eukaryotes, it's a little more complicated. That was prokaryotes. Like so many things, we're a little more complicated. We have in the order of 9 or more initiation factors, so it's not quite as simple to assemble a functional ribosome. The initiator methionine is just simply methionine. It's not formal methionine. So you do need an initiator, specific initiator tRNA with methionine on it. And you still need messenger RNA, of course. But there's some other interesting things as well. So there's a cap binding protein. You may remember that we modify messenger RNAs by adding on that backwards added guanine with a methyl group on it, so it kind of looks like it has a 3' end. That's the methyl G cap. And there's a cap binding protein that has to be bound to that cap for messenger RNA to be used. There are helicase activities because the messages in eukaryotes can be a lot bigger than the ones in prokaryotes, and you may need to unwind them enough. There's a poly-A binding protein. Excuse me? Poly-A binding protein. Poly-A tail is down at the 3' end. What is it doing at the initiation site? It's turning out to be very important. Bear with me. There's an exciting surprise at the end. So the poly-A binding protein bound to a poly-A tail can interact with the eukaryotic initiation factor. And so this means that a functional eukaryotic message is a circular molecule with the 5' end with its methyl G cap kissing and hugging with the 3' end to make a circular molecule. This turns out to be important for quality control. So now we have this circular molecule. The 40S subunit, the small ribosomal subunit for eukaryotes, is going to actually use up energy to scan the messenger RNA. It's going to ride along the messenger RNA looking. It's looking. And it's going to look for basically the first GCCA or GCAUG. So that's the magic AUG. That's codes for methionine. There's this context around it that says, this is the AUG you want to use. And when it finds that particular AUG, initiation factor E for eukaryotic, EIF2, can again deliver tRNA to the P site. Again, GTP is hydrolyzed, and that liberates the EIF2. And now the 60S subunit can come in, and we have a complete ribosome ready to go. So some extra complexity, but the fundamental process has a lot of similarities to the prokaryotes. So now we have an assembled ribosomal complex. We're ready to elongate. If we've got a protein that's 300 residues long, we're going to have to do 300 additions of amino acids. And there's large similarities in each case. So in every cycle of elongation, there's going to have to be an amino acyl tRNA come in and bind at the A site. So it's going to come in, and if it's complementary to the codon, it can be delivered there, but it won't just simply float in. It's delivered by a GTP binding protein called EF. EF here for elongation factor, and it's EFTU in E. coli. TU turns out to be for temperature unstable, in case you're ever curious. EFTU is a very powerful little protein. It recognizes all tRNAs. All tRNAs look alike to EFTU. And so it can recognize things like the TFC stem and the acceptor stem. So these are common shared features of all tRNAs, and EFTU will accept anything and bind to it. But it is a GTP binding protein. Now, whether or not a tRNA with EFTU bound to it will actually bind at the A site is going to depend on the codon there, and that the codon is going to determine whether it has the affinity that's needed there. This is kind of an interesting diagram because it gives you a sense of just how tight the space is on a ribosome, that the ribosome actually has to kink the messenger RNA a little bit, to bend it a little bit to make the bases splay out a little bit so there's room for 3 tRNAs to fit in there. The P site was filled by the initiation factors. Here's the A site filled by EFTU. Here's the E site for the tRNA that's about to leave. And the part of the job of the ribosome, of the small subunit, is to check this and to see that that's as good a fit as it can be. Now, we've talked about proofreading during DNA synthesis, a little bit about RNA synthesis. There's proofreading in protein synthesis to some extent as well. And it's been called kinetic proofreading. That if you have an EFTU delivering a tRNA that's correct for that codon that's exposed, the correctness of the fit will signal to the EFTU that this is a good fit. EFTU will hydrolyze the GTP and then go away. And that leaves then the amino acyl tRNA charge sitting at the A site. If they don't fit properly, you know, they may bang around for a while, but if they don't fit properly, there won't be GTP hydrolysis, and the complex of EFTU and tRNA will float away and try some other ribosome somewhere else. This means that the protein synthesis process has an error rate of about 1 in 10 to the 4th. It's not too bad. I'm not that good. Not as good as DNA synthesis. But of course, proteins don't live as long as DNA does. And this is showing you that cycle in a sort of a diagrammatic form. Here you have a ribosome that has on it a tRNA in the P site. And it's the P site because here's the peptidyl chain growing. There's 4 residues there. You have a charged EFTU with an amino acyl tRNA on it. And this particular one isn't quite the right one. So it sits down for a while, but it comes off. And it may go through a number of cycles of that. But if an EFTU with the correct amino acyl tRNA on it comes in and sits down, the correctness of that fit signals to the EFTU to hydrolyze the GTP. It comes off, and it leaves behind the ribosome now with a peptidyl tRNA in the P site, a charged correct amino acid in the A site, and it's ready now for the next step called transpeptidation. What this means is it costs a GTP per amino acid for accuracy. You can imagine in principle that it could just float in and leave it to chance. Having EFTU hydrolyze the GTP, it means that the cell finds it worth spending a high energy bond per amino acid to check the accuracy. And this ribosome here, with both A site and P site occupied, are ready to go. They're not showing you the E site here. They often don't. So we have a ribosome messenger RNA complex with both the A site and the P site charged. We really want to make a peptide bond. We want to actually elongate this chain. And this work is going to be done by the 50th subunit, the large subunit. So the small subunit was busy looking at the RNA and fitting it. The large subunit is doing stuff up at the other end of the tRNA. And actually, one thing that's nice about this diagram is it kind of represents that in terms of having, stuff down at the bottom and stuff at the top. Interestingly enough, making that peptide bond through the activity called peptidyl transferase does not need any more energy put in. If you remember, we had this amino acyl tRNA. This bond looks like it should be a reasonably high energy bond, and in fact, it has enough energy to do the next step. So what's going to happen? You have the chain here so far. Here's the charged tRNA, the amino acyl tRNA in the A site. We want to make a bond between this nitrogen and this carbon. That's the peptidyl transferase activity. And when that transfer is accomplished, we now have this full chain in the A site because this growing chain got smooched onto that nitrogen. So there's an empty tRNA in the P site at the moment, and the peptidyl tRNA for the moment is in the A site. So the growth of the chain is from the amino terminus to the carboxy terminus. You can see here's this chain had a free amino terminus just like this tRNA does. This is the carboxy end. So we're adding the N-terminal amino acid as the first one to come on. The C-terminal amino acid is the last one to come on. And interestingly, to catalyze that reaction, to smush those two together, that is catalyzed by RNA in the large subunit. This is an example of an RNA catalyst, and it's at the very heart of life that every ribosome uses RNA residues as their catalyst. And we see our old friends, proximity and orientation. Two of the things that the large subunit does is to hold them close together and to hold them in the right position. And that combined with the energy inherent in the amino acyl bond is enough to let it happen. Now, we can't leave the ribosome like that or things would come to a halt. We're going to have to move that new peptide tRNA down over into the P site, right? We're going to have to get the growing chain out of the A site into the P site so we can do this again. So we're going to slide the tRNA into the P site of the ribosome. The empty tRNA, the one that used to be have the peptide chain, it slides into the E site, the exit site. And the whole messenger RNA slides down one codon. So the ribosome can kind of ratchet it down one to keep it in frame, so the next codon is now exposed. I did it right this time. Remember, cough into your sleeves, please. Sorry about that. Now that's a lot of tricky stuff to do. And it turns out that there are elongation factors that assist with this. EFG, kind of a funny name, but there it is. So elongation factor G is the elongation factor responsible for this. And it interestingly looks kind of like EFTU with a tRNA on it. So it's kind of a molecular mimic of EFTU when it's charged. And it seems to be able to kind of bump that growing peptidyl tRNA down one. And EFG, like EFTU, has a GTP bound. When it's accomplished this sliding motion, it hydrolyzes its GTP and then it leaves. And it might be useful to see that whole cycle all in one diagram. This is the elongation cycle for protein synthesis. We set up an initiation complex, and then we're going to elongate it through this cycle for every single residue. And so here we have a ribosome that's got a P site occupied with a peptidyl chain growing in it and an empty A site. EFTU delivers the tRNA, hydrolyzes its GTP, and goes away. So now we have a messenger RNA fully occupied in the A and the P site. The small subunit helping to check on that codon. Transpeptidation happens. That's the transfer of this peptide chain to this amino acyl group. So you can see now that the growing chain is in the A site and we have an empty tRNA there. We need to shift stuff down. So the translocation is EFG with GTP on it, can sort of sit in there and kind of look like it's the right thing. When it hydrolyzes the GTP, it comes off, leaves the empty A site, and now we can do it again and again until every codon has been read and turned into a protein. So GTP does a lot of things here. We saw it was doing it back at the initiation step. It's involved in the delivery of the tRNA and the proofreading step there. GTP is involved with the translocation step. It's only this step here, over at the transpeptidation, that the inherent energy of the amino-acyl bond is sufficient to accomplish the job of catalyzing that reaction. So it costs us 2 GTP for each and every peptide bond. Any questions? Okay. So we've done this 50 times, 100 times, 1,000 times, depending how big the protein is. And we have to come to the end at some point. There has to be an end of the protein. And there are codons. I don't want to jump back at this point, but if you go back and look at that table of codons, you'll notice that there are 3 codons in it that are called stop. They don't code for an amino acid, they code for stop. And so any one of those 3 codons can be used to bring to an end the process of elongation. And so this elongation cycle continues until you get a peptidyl tRNA sitting in the P site, and now next to it, the messenger RNA is slid down, and you have there a stop codon, one of these 3 codons that do not code for a protein. There is a release factor, RF, 1 or 2, that can recognize that as a empty stop codon. And the release factor tricks the ribosome into transferring the peptide chain not onto a tRNA, but onto water. And so the peptide is hydrolyzed off of the tRNA and now free to go and do whatever it's going to do. That still leaves you with a tRNA sitting at the P site and a release factor sitting at the A site. A third factor comes in, release factor 3. Release factor 3 is another one of these GTP binding proteins. And release factor 3 can catalyze the release of the RF, the release factor, out of the A site. This is still not a completely free ribosome. There are other factors involved in disassembling the empty ribosome and getting the messenger RNA out of the way. This is a picture of RF of a eukaryotic release factor. And in the eukaryotes, it's RF1 that has a GTPase. But release factors kind of look like the anticodon end and acceptor ends of a tRNA. So it's, again, a molecular mimic for some of the stuff that would normally be involved in elongation. And so there's a 3 amino acid sequence at one end of it that mimics an anticodon. And maybe that's a good place to stop for now. Okay. Hello? AV person? Oh, I hate doing this alone. I hate doing